In this part, we'll be discussing about the megasporogenesis, that is the development of megaspores. This picture is clear to us. It is nothing but a longitudinal section of the female cone where it shows the number of megasporophylls present on either side of the central axis. Each megaspore usually contains two types of scale, that is ovuliferous followed by this bract scale. This is an enlarged view of the same one. There is a single megasporophyll has been enlarged here, where the things like this is the ovuliferous scale followed by the bract scale are clear, and there is a vascular bundle also showing this applied to bract as well as ovuliferous scale. Followed by we have this micropyle. Actually this is the development of the ovule, where formation of the micropyle followed by the pollen chamber, where pollen grains are going to rest prior to fertilization, and integument, that is a covering of the ovule, followed by nucellus region and the formation of the embryo sac. This is what we are going to see now in a more diagrammatic view. So the hypodermal cells in nucellar tissue at the micropylar end is differentiated into archisporial cells. So we have this nucellar tissue, right, at the micropyle end. So this will be the micropyle end. So the nucellar tissue near this micropyle end is going to differentiate as what? Archisporial cell. Now these archisporial cell, they are going to divide periclinally. So they are going to divide, this will be the archisporial, it is going to divide periclinally, leading to formation of the two cells, upper parietal cell and the lower megaspore mother cell. Archisporial cell, it underwent periclinal division leading to formation of the two cells, upper parietal cell and the lower megaspore mother cell. Now this upper parietal cell, it is going to divide and form what is called as tapetal layer. This tapetal layer is nothing but tapetum, which is going to provide nourishment or nutritive, rich in nutrition. It is going to provide nourishment to the growing megaspore mother cells. Here these megaspore mother cell are going to take nourishment from this tapetal layer. These megaspore mother cell undergo meiosis leading to formation of the 4 haploid megaspores. Among them 3 are going to degenerate, which are towards the micropyle end. They are going to degenerate, and remaining 1 will be the functional megaspore. Now the outer 3 megaspore degenerated while the lowermost megaspore becomes the functional one. This is how it is going to look like. The upper free opening of the integument, this one, it is going to form, it will be like open here, open end. This will be the micropyle, and a small space or a concavity is present between whom? Between the integument and the nucellus. That is nothing but a pollen chamber. This one they have not mentioned here. This pollen chamber is nothing but a space where the pollen grains are going to come and rest. And after pollination, the pollen grains are stored in the pollen chamber, and further development of pollen grains takes place in the nucellar region. So this is the nucellar region. They have directly shown the formation of the pollen tube. This pollen tube are going to grow in this nucellar region. These are the archegonia, where a female gamete, that is egg, is present. Endosperm is nothing but, we will be discussing regarding this one. Endosperm is nothing but a free nuclei division, which is again a nutritive tissue required for the development. This is the female gametophyte, nucellus, megaspore membrane. So this was a description regarding the megasporogenesis in case of Pinus. Thank you.